So I think Daniel Haber sort of talked to you about sort of the work that I've been doing on pancreatic circulating tumor cells with sequencing. So I'm going to be telling you about another story that happened during this project. And so here's our fancy, very expensive machine that we had talked about before. Basically, our intent was to do CTC RNA sequencing, as well as primary tumors, normal tissues, and sort of metastatic sites, and that was our primary intent for sequencing. And this is the mouse model that was developed by Nabil Bardisi and Rhonda Pino's lab, and this is the model that we used, again, sort of highlighting what Daniel had shown. But the bottom line is that when we sequence these primary tumors from the mouse, primary pancreatic ductal adenocarcinoma, as well as some normal liver in the same mouse, and we looked at usable reads. So these are total transcripts of RNA that we were sequencing here. And then we align them to the known transcriptomes, the known space of RNA in the universe. What we found was, only 11% of our reads in this tumor were aligning to the known transcriptome, while in the liver and in a pancreatic cancer cell line that was similar, we saw like a 49 to 62%. This is sort of what you sort of see with next-gen sequencers. So, initially we're like, well, is this true biology? Is this the pancreatic tumor, or is this just is the machine broken? And so we looked and said, it must be coming from the genome. So then we said, let's take all these reads and align them to the genome. And lo and behold, there's all these reads that 59% now align to the genome, while these stay about the same, comparable in the 50 to 60% range. So this indicated that there's a huge amount of stuff that's coming from the genome, but that's not annotated in the known coding RNA. So that was very interesting. And so what is this RNA? And so here's a histogram plot of all the helicose reads. But those black bars are basically showing where the helicose reads are aligning. And you could see here, okay, well, let's do this one. So you can see here that all these reads are piling up here. So the interesting thing is here, if you look at ref-seq, so these are known RNAs, there's nothing here. More interestingly, if you look at where all these reads are in terms of conservation across all other species that have been sequenced, there is zero conservation. That's very an interesting thing. So, so I looked at this. There's some cDNA stuff here. There's some predicted stuff. But the bottom line is it was highly repetitive. There was reads in both directions. It didn't seem like it was polyadenylate based on poly A enrichment followed by sequencing. And what we ended up finding was it was this stuff down here, this stuff in this thing called repeat masker, the thing designed to mask the repetitive elements. And what we found was that this was, in fact, the major satellite repeat. And so what is the major mouse satellite repeats? The satellite repeats are these massively repetitive sort of tandem arrays found mainly in the centromeres and telomeres of chromosomes. In the mouse, this is an acentric sort of chromosome in the mouse, and the major satellite repeat is showed here, and the minor satellite here, and sort of is combined in this sort of centromeric area. This is about 3% of the entire genome. It's pericentromeric, and it's sort of controlled by epigenetic marks H3K9, as our epigenetic experts here know. And so this was what we knew, where this particular thing was coming out. So we looked at other tumors. So these are more additional pancreatic mouse genetically engineered tumors with KRAS and P53 loss. We also looked at KRAS and SMAD4 loss, and we looked at Kevin Hagis's colon mice as well as Kwok Kwan's lung cancer mice. And what you can see here is that we have lots of satellite reads in all these tumors. Interestingly, cell lines derived from these same tumors pretty much had very minimal amounts of satellite reads, and normal tissues had very small amounts. And if you look here, this first mouse here, 50% of all RNA was coming from the satellite, from the satellites, which is a huge amount. And so when you look at that, just sort of average all of them, all the primary tumors, 10% of all the reads of RNA coming from the primary tumors in these mice were coming from the major satellite repeat. That sort of rivals ribosomal RNA here, and this is mRNA here. Normal tissues, pretty much did not have any of the major satellite repeat, and you can see here the transcriptional expression is completely different. I think the thing to highlight in this particular slide is as well, there's all this 47 to 50% of reads that are unannotated, i.e. they're aligned to the genome, but not known to be in the known transcriptome. So this offers a huge area of discovery for the future. So to validate this, we sort of developed a northern blot probe. And here, the northern blot, you can see here in 3 independent mouse primary tumors, you have this huge smear. So it's not one band. It's a huge amount of satellite transcription that's occurring that's either being processed or is produced at various sizes. And so if you take this initial tumor here and then generate a cell line from that, you see that these satellites are now gone. However, you can re-express those satellites when you treat them with a hypomethylating agent, 5-azacitidine, and you can see the satellites go from not being present to present in adherent culture. And then more interestingly, if you look at the cell line, again, grown in adherent culture in vitro, there's nothing there. But when you take that same cell line, put it into a nude mouse, and generate a xenograft, satellites come back. So indicating that this non-coding RNA is specific for in vivo tumorigenesis. And so we then applied this northern blot probe to RNA in situ. And so in the RNA in situ, the counterstain is eosinogen. So this is pink is negative, will counterstain the tissue, and purple is the positive for satellites. And you can see that the primary tumor is a huge amount of satellite expression by RNA in situ hybridization. And when we look at these mice, they actually have Mets. And so when we look in the liver, there's no signal of the satellite in the liver, but you see that this large glandular macromet is lining up for the satellite. And more interestingly, you can sort of see like these 2 or 3 cell micromets that sort of light up with this satellite, suggesting that this possibly could be used as a biomarker. The other thing that we found that was very interesting is that it was present at early sort of Pannin stage of development in this particular mouse model. So from the Pannin stage, you sort of see satellites. And as you get sort of the higher high-grade Pannin with these papillae, you see even more satellites. So this suggests that this marker is a very early marker in tumorigenesis and provides a potential early diagnostic marker for pancreatic cancer, which is sorely needed. So the obvious question is, we're not here to cure mice, so let's look in humans. So we looked at 15 pancreatic ductal adenocarcinomas from the MGH pathology department, looked at normal pancreas, and we looked at a variety of different other cancers, so non-small cell lung cancer, renal cell cancer, ovarian cancer, and 3 prostate cancers. And then we also sequenced a bunch of normal tissues like brain, colon, kidney, lung as well. And you can see that satellites are produced not very highly in normal pancreas, but in other normal tissues as well as in cancers. So then when we looked at the satellites, we found that in the human there's 25 different satellites, while in the mouse there's about 2 or 3. So in the human, we found that there's particular satellites, if you sort of look at it in terms of black is sort of high in cancer, and white is high in normal tissues. You can see that certain satellites are being preferentially expressed in tumors, while certain ones are being expressed preferentially in normal tissues, which is very interesting from a biological standpoint, because it indicates that these repetitive elements are differentially expressed in cancers over normal tissues. And one in particular was found to be massively overexpressed in tumor cells when you look at fold comparison. So in the cancer cells, these were like basically 130-fold higher in expression compared to normal tissues. And so when you plot that out, you can see here that actually all 15 of these have HSAT2 reads, while normal pancreas had 0 reads of HSAT2. All these cancers were as well positive, and none of the normal tissues except for fetal brain and a little bit in prostate had a little bit of HSAT2, indicating this is a highly specific repetitive element that's being expressed in primary tumors of human. So we then worked with Panomics, which is now Affymetrix, on an RNA in situ hybridization technique that we could use in FNA biopsies in human specimens. And in this particular specimen, this is the H&E over here, and over here is the satellite stain. But you can see the satellite stain are the red dots, and those are the indicative of HSAT2. And so we see that this particular satellite is being expressed at high levels in the tumor and not so much in sort of normal adjacent sort of leukocytes. And just as we saw in the mouse, these pre-neoplastic pannin in the human, I don't know why that is there, but there's, I must have been thinking about Sid Mukherjee that day. In pre-neoplastic pannin, there's a lot of satellites in the pre-neoplastic pannin. So what do satellite overexpression really mean in cancer? I'm just sort of going to gloss over this very quickly for time, but what you can do is you sort of look at all the other genes and sort of look at ones that are linearly correlated to the satellite expression across all tumors. And so we sort of defined a set of highly correlated genes of an R value of 0.85 or higher as satellite correlated genes. And so when I did this, I sort of looked at these genes. I said, oh, look, there's a bunch of them. 63% of them are certain neurals. Some of them are sort of enriched in stem cells. We got HOX, we got zinc finger. And I remember this phenomenon that I had seen in clinic this was at the Farber, not at MGH, but where a prostate adenocarcinoma had actually turned into a poorly differentiated neuroendocrine tumor. So I said, maybe this is neuroendocrine differentiation. And so when you actually look at IHC for chromogranin A, which is sort of a classic neuroendocrine marker, you see as the percentage of satellite being expressed in the tumor, you sort of get more and more protein for the classic neuroendocrine marker chromogranin A, indicating that neuroendocrine differentiation is sort of associated with higher satellite expression. And this would be relevant in neuroendocrine differentiation of the prostate, as well as that some of the stuff that Jeff Engelman had showed today, showing that maybe non-small cell lung cancer with the EGFR mutations turn into small cell lung cancer. And in the human, it's, we didn't have FFPE blocks for those ones that we had sequenced, but it looks like it's not going to be chromogranin A, but these other sort of human neuroendocrine markers that seem to be differentially expressed in high satellite versus low satellite tumors. So obviously, from a diagnostic standpoint, since I worked on CTCs, the obvious question is, can we use satellites in pancreatic CTCs? And this is the chip that we had used for the pancreatic CTCs, and Daniel had already shown this. This is sort of standard IF for cytokeratin, EpCAM staining, and CTCs in human pancreatic cancer. We successfully captured human pancreatic cancer in patients at the MGH Cancer Center. So the question was really, can we apply this same HSAT2 technique to CTCs? So this is very early data, but the early data suggests, and here this cell's about 20 microns long. This nuclei is fairly large, this prominent nucleoli here. You see the HSAT2 signal. In this case, in the fluorescence mode, it's orange. We are detecting a lot of these cells in metastatic pancreatic cancer patients. We still don't know what exactly those cells are. However, it's suggestive that they may be circulating tumor cells, and this particular technique may be more sensitive than standard keratin markers for identifying CTCs. And early data suggests head-to-head versus IF cytokeratin is that we may have a 10-fold improvement in CTC detection with this technique. So the bottom line is satellites are expressed in massive amounts in a wide variety of epithelial tumors, but not in normal tissues. They're expressed at a very early stage in tumorigenesis, indicating that they could be an early detection marker. And if you have a blood-based one, then you have a blood-based screening test. Satellites are repressed in cultured cell lines in vitro, probably through some epigenetic changes, but can be induced in vivo, suggesting that this particular thing is set off in vivo, and really highlighting how in vivo tumors probably behave very differently compared to cell lines grown on tissue culture plates. And ultimately, it's my dream to sort of use this sort of probe as a way to identify CTCs better in pancreatic cancer patients, because we really need some type of early detection screen for this horrible disease. And that's it. So I'd like to acknowledge Daniel Haver and Shama Swarns, who have been my co-mentors. Many people have helped in this project, and it's been a great ride. And between this and the Win2 project, Daniel has me very busy.
Can you measure these repeats, not in CTCs, but just on genomic, looking at DNA in the blood?
Right, so like serum-free nucleic acids. So right. We've tried to do some of that. It's a little challenging to do that with sort of ish techniques. Obviously, there's no cells, but when we did try to sequence serum or cell-free nucleic acids in the serum of patients, and we see something there. It's not so clear exactly what we're seeing, because we do see a lot of satellites, but it's not certain that that particular modality will tell us cancer versus not. So it's something that we had initially looked at, but right now we're sort of CTC centric. So
thank you. Comments, questions, please.
Are these satellites ever expressed anytime during normal development, or?
Yeah, great question. So I didn't show you, but we did an RNA-ish of total embryo slices of the mouse. We also did a northern blot in the mouse. It is seen in embryonic development. So embryonic day sort of 6, 8, 12, 15, we see it. The interesting thing is it looks different. It's a high molecular weight on northern blot, while in the tumors you saw there was a big smear. So there's some difference in that processing. In the embryos, we see that there is some expression in the fetal liver and the fetal lung. And those are the only two sort of major organs that we see it in by RNA-ish. But we have done that, and clearly the two are linked, embryogenesis with tumorigenesis, I believe. We're not sure. We don't think so. So when you do like a poly T25 sort of enrichment to look at, just trying to get all the poly A tailed RNA and then sequence it, you don't see it there.
Do you have any sense of what regulates their stability in the absence of polydemylation, or?
No idea. But I think the interesting thing is that it's so repetitive. So like these HSAT2, you'll see like 25 base pairs over and over and over again, and they're huge. You see, if you look at the northern blot, at least in the mouse, they're on the order of 10 kb sometimes, and they're sort of fragmented all the way down to the 100 to 50 base pair range. I think there's some element of sort of RNA processing that's involved, linked to epigenetics, which I think our epigenetic colleagues can sort of tell us more about because they are the experts.
Do you have any sense how these correlate with prognosis in pancreatic cancer, for example?
In pancreatic cancer, well, unfortunately, Bruce is laughing, but he knows that my answer, which is in pancreatic cancer, prognosis is all bad. So unfortunately, all comers, we only have a 5% cure rate of all comers with pancreatic cancer. So whether or not it's prognostic or not is sort of irrelevant, unfortunately, in pancreatic cancer. However, what I can tell you is that sequencing of those 3 prostate cancers, the highest sort of satellite producing prostate cancer was a Gleason 9 prostate cancer. So it suggests that there may be some connection to grade, but in pancreatic cancer, all grades are bad. So.
Okay, thank you very much, David.